Alright, this is Jahangir from MedPorium, and today we have part 2 of our lymphatic and immune system unit, in which we'll discuss innate immunity. Let's first begin this discussion by defining what innate immunity is. Innate immunity is a non-specific system of defense always prepared to fight a pathogen. Now within the immune system, we have 3 defense lines against foreign pathogens. The first includes the first line of defense. The first line of defense consists of external barriers such as our skin and mucous membranes. The second is known as a second line of defense. This is our non-specific defense and is initiated when a pathogen breaks through external barriers, in which leukocytes, macrophages, and antimicrobial proteins work to defend the body. The third and final line of defense is known as the third line of defense. The third line of defense is based on memory and it is employed when the body encounters a pathogen previously fought. You can think of the third line defense as a body putting a wanted poster on a specific pathogen in which our lymphocytes will attack that pathogen. One thing to note is that innate immunity includes our first and second lines of defense. Adaptive immunity, which is the second type of immunity, includes our third line of defense. Let's understand the first line of defense more thoroughly. As we discussed, the first line of defense includes external barriers such as our skin and mucous membranes. Now our body will be exposed to many different pathogens from the Staphylococcus bacteria to the Streptococcus bacteria. Let's say we're exposed to the Streptococcus bacteria. In this scenario, we touched a file cabinet that contained the Streptococcus bacteria. Here's what that may look like. Now the skin is waterproof and usually impenetrable for bacteria to enter the body. Sweat coats the skin with a protective acid mantle, which is a thin film of lactic acid that stops bacterial growth. Sweat also contains the antimicrobial peptide known as dermcidin, which is toxic to bacteria. In this scenario, however, we try to cover our cough with the hand containing the Streptococcus bacteria. Now the bacteria has moved on into the upper respiratory tract. This is where mucous membranes come into play. Now our mucous membranes have a thick consistency, limiting the movement of the bacteria. Mucus and saliva contain lysozymes, which destroy bacteria. Our lungs are covered with a thin mucous membrane as well, including a structure known as cilia, which pushes foreign pathogens out of the lower respiratory system to be coughed out later. Our immune system is always in war, preventing a pathogen from causing harm to the body. However, in this scenario, the Streptococcus bacteria has successfully passed the first line defense and has entered the lower respiratory tract. Now what happens? This is when the second line of defense is activated, and this includes our leukocytes and macrophages. There are 5 different leukocytes. The first includes neutrophils. These are the most common white blood cells in that they like to phagocytize pathogens. The neutrophils are special in that they perform the action known as respiratory burst, which is the release of many toxic chemicals in the phagosome that ends up killing the neutrophil itself. The second includes basophils. Basophils secrete leukotrienes, which bring the attention of neutrophils and eosinophils. They also secrete histamine, which is an inflammatory chemical, and heparin, which promotes clot formation. The third includes eosinophils. These are the most concentrated in sites of allergy, inflammation, or parasitic infection. They phagocytize pathogens much like the neutrophils and secrete enzymes that limit the action of histamine and other inflammatory chemicals. The fourth includes lymphocytes, which we discussed in part 1, and the fifth type of leukocyte includes our monocytes. Monocytes are created in the red bone marrow, as we discussed, and mature into macrophages once they enter the bloodstream. One thing to note is that neutrophils, eosinophils, and basophils are part of a group known as the granulocytes, which are cells with small granules in the cytoplasm. Lymphocytes and monocytes are part of the agranulocyte group, since they do not have small granules within the cytoplasm. The leukocytes listed, as well as our external barriers, will release antimicrobial proteins to fight off a pathogen directly. There are 2 main antimicrobial protein families. The first includes interferons. Interferons are signaling proteins secreted when a cell has been infected, much like the cell's dying words, in which nearby cells will be alerted of a foreign pathogen. Interferons bind to the surface receptors of a nearby cell to bring cellular immunity from a particular pathogen. The second antimicrobial protein family is known as a complement system. The complement system is a group of plasma proteins that aid in non-specific and specific immunity. These proteins are synthesized by the liver and are active when the pathogen enters the bloodstream. The proteins are usually represented by a C, then a number. There are 3 different routes in the complement system. The first is known as the classical pathway. Now, the classical pathway is antibody dependent, meaning that there must be antibody binding to an antigen of a foreign pathogen. The protein C1 or C1 complex, if you will will bind to the antibody. C4 will come in and be broken down to C4A and C4B by C1 complex. The same process occurs for C2, forming the C4B2A complex, which is also known as the C3 convertase. The second route is known as the alternative path. This is an antibody-independent mechanism that occurs when C3 experiences hydrolysis, creating C3H2O. Factor B, which is another protein in the complement system, will cleave into factor BB and factor BA by factor D. Factor BB will bind to C3H2O, forming the C3H2OBB. C3H2OBB will cleavage C3 into C3A and C3B. The C3b will attach to factor BB, forming the C3b-BB, which is the C3 convertase of the alternative pathway. The third route is known as the lectin pathway. This pathway is activated when the mannose-binding lectin, also known as MBL, binds to a carbohydrate, usually mannose, which is found in the microbial cell surface, in which mannose-binding lectin serine protease 1 and 2, or just MASP1 and MASP2, will bind to mannose-binding lectin, forming the C1-like complex. For simplicity, we will call this the MBL-MASP complex. The MASP2 from the complex will cleave C4 into C4A and C4B, and the same process is seen for C2, creating the C4B2A complex, which, if you can recall, is our C3 convertase. Another molecule known as ficillin will have the same functions as the mannose-binding lectin, but ficillin can bind to different types of carbohydrates. In the complement activation, the C3 convertase created by the three pathways will cleave C3 into C3A and C3B. This produces four different end results. When C3 dissociates into C3A, C3A influences mast cells and basophils to secrete histamine and other inflammatory chemicals, which activates the action of neutrophils and macrophages to inflame the tissue. This in total creates inflammation. On the other hand, C3b binds to the antigen-antibody complex, in which red blood cells transport that complex to the liver and spleen, where macrophages will phagocytize the complex. This process is known as immune clearance. C3b can also coat the pathogen in a process known as opsonization. Essentially, C3b makes the pathogen tastier for neutrophils and macrophages, promoting phagocytosis. Lastly, C3b will bind to the C4b2a complex, creating the C4b2a3b complex, which is the C5 convertase. This convertase will split C5 into C5a and C5b, in which C5b will bind to C6, C7, C8, and C9 to create the C5b 6, 7, 8, 9 complex. This complex is known as a membrane attack complex, or just MAC for short. The membrane attack complex will attach to the pathogen to form a hole to expose the pathogen to the extracellular fluid, in which the pathogen will die since it cannot maintain homeostasis. This process is known as cytolysis, which is the most significant action from the complement system. Here's the end result of the cleavage of C3. Let's discuss another mechanism the body employs to limit the damage a pathogen may cause, and that mechanism is known as inflammation. Inflammation is a local defensive response in an injured tissue to 1. Limit the spread of the pathogen and destroy it, 2. Remove debris of the tissue, and 3. Repair the damaged tissue. There will be 4 signs of inflammation. There will be redness, swelling, heat, and pain, usually associated with the tissues of the skin, which is known as cutaneous inflammation. There will also be a loss of function. Inflammation usually occurs from physical trauma, toxins, or the entering of pathogens. So let's just say you accidentally step on a dirty nail. That nail has now pierced through the epidermis into the dermis layer of the skin. At this point, the nail has passed the first line of defense, which is your skin. The nail will most likely have bacteria such as the Staphylococcus bacteria, which will enter the dermis. Let's understand the inflammatory response, starting with the vascular response that will occur during inflammation. Now, the nail will also have damaged the cells of the epidermis and dermis, resulting in the release of chemical mediators such as histamine, kinins, and prostaglandins, which promote vasodilation, hyperemia, which is excess blood flow, increased capillary permeability, and local edema. Vasodilation and an increased capillary permeability are responsible for redness, heating, and swelling, while edema is responsible for pain. Now let's discuss the cellular response. Neutrophils and macrophages will circulate the bloodstream, and when chemical mediators from damaged cells are released, this attracts the leukocytes to the area of the injury. Mast cells, similar to basophils, will also secrete their chemical mediators, initiating the inflammatory response. Hemotaxis, which is a directional migration of white blood cells due to the inflammatory chemicals, will be initiated, as well as margination, which is the process of leukocytes adhering to the vessel wall due to cell adhesion molecules known as selectins. The leukocytes will then perform diapedesis, which is the process of leukocytes squeezing through the endothelial cells. They are able to do this due to increased capillary permeability. At this point in time, our leukocytes have entered the site of injury and perform phagocytosis. The complement system will also be activated throughout this invasion. Leukocytes will secrete cytokines to bring more leukocytes into the area of injury. Chemotaxis, margination, and diapedesis will continue on and on until there are more leukocytes, enough to dismantle that specific foreign pathogen. By this time, hopefully, the pathogen has been dismantled. Macrophages will continue to phagocytize cellular debris from the immune response. Our lymphatic capillaries will influence an increased lymphatic flow due to the high interstitial fluid from edema in which additional debris will be collected. Then neutrophils and macrophages form a creamy substance known as pus. Platelets and endothelial cells will secrete platelet-derived growth factor, which stimulates fibroblasts to multiply and synthesize collagen to repair the damaged tissue. Hyperemia will bring oxygen and amino acids, which speeds up tissue repair, while heat increases metabolic rate, speed of mitosis, and tissue repair. The last mechanism that is part of the innate immune system is known as a fever. The fever is something we've all experienced, and one of the common symptoms includes hyperthermia. The fever is a temporary but beneficial resort in fighting a pathogen because 1. It promotes interferon activity, 2. It elevates metabolic rate, which means faster tissue repair, and 3. It stops the reproduction of pathogens. The fever is initiated when neutrophils and macrophages secrete endogenous pyrogens when fighting a pathogen, in which the hypothalamus will raise the temperature of the body. The fever ends when the endogenous pyrogen secretion ceases, in which the hypothalamus will lower the body temperature back to normal. Alright, and unfortunately that wraps up part 2 of our lymphatic and immune system unit, in which we discuss innate immunity. In this discussion, we discuss the 3 lines of defense of the immune system, the 5 different leukocytes, interferons in the complement system, inflammation, and the fever. And as always, a Pomodoro day make you doctor someday. I'll see you next time.